Hello, everybody, and welcome to our 25th installment of the PhenoTips Speaker Series. This is quite a landmark installment here, so I'm very excited that we're going to be diving into population genomics in clinical practice today. I'm your host, Kira Deneen. Our panelists are amazing. I've really been looking forward to this for weeks, I have to say. And what's very cool about our panelists is they each come from a different country. So we got the US represented, the UK, and Canada. So I think that's going to provide for a very interesting conversation just with how different healthcare systems are working within this context of population genomics in clinical practice. So thank you for everybody that is trickling in. We really appreciate you taking time out of your day to be watching this live, or if you're listening to this later as a podcast. So we really appreciate you just taking the time and energy to hear our conversations here. The way this is going to work today is I'm going to be interviewing our amazing panelists, but I want you guys to submit questions. So if you're watching this live, we would love to have you submit questions. We have the Q&A box there, so you can put questions directly in there. Don't wait till the end, though. I will probably wait till the end to actually ask your questions to our guests, but I don't want you to think about it and then say, oh, I forgot what I was going to ask or got busy or, doing something else while you're listening. So please, as you have questions, pop them into the Q&A chat. I would love to be able to have, our three doctors here answer those questions. So a little bit about PhenoTips, who is the sponsor for this series. They've been sponsoring the series since 2020. And as I said, this is our 25th installment. So we have 24 other awesome episodes, installments that you can go check out on demand. That's at phenotips.com. So PhenoTips, for those that don't know, is a complete genomic health record with searchable, shareable, structured, and scalable database of patient records that are also linked to family records. So the PhenoTips genomic health record includes tools like inclusive pedigree builders. We have a really, really good installment specifically about designing and making sure you're asking questions to be inclusive with pedigrees as you're getting that information. They also have the human phenotype ontology capture, digital patient questionnaires, built-in risk assessments, and diagnostic insights. So it really is this complete suite for genomic medicine. And a lot of us have experienced, I'm sure the four of us on this call here have experienced that electronic health records are not built for genetics. Usually there's a little section to it and it doesn't really flow in nicely. It's kind of an add-on. So that's why PhenoTips really saw this need. They wanted to close that gap by providing this genomic health record and just providing that unified and seamless genomics workflow. So it's really cool. Check it out if you haven't yet. And as I said, my name is Kira. My pronouns are she, her. I'm your host for this webinar for people that have are just joining in now. I also am the host of DNA Today, which is a multi-award-winning genetics podcast. We have produced over 250 episodes in the last 11 years, and they're very similar to conversations that we have on the PhenoTips Speaker Series here. My other role is as a prenatal genetic counselor. So from a clinical perspective, that's where most of my experience comes from. But enough about me. I would love for our three guests here to introduce yourselves, because people that are listening to this in the podcast format don't get that visual of who's talking. So I think it's helpful to hear whose voice is who at the beginning. So Dr. Green, I would love for you to introduce yourself of what your title is and what your role is in the NIH.
Sure, happy to be here. Thanks for the opportunity to spend some time chatting with you and these wonderful other guests. So I'm Eric Green. I'm director of the National Human Genome Research Institute at the U.S.'s National Institutes of Health. So I've been at this institute now for 29 years. I've been the director for nearly 14 years. My role, we're one of the 27 institutes at NIH, but we're the ones responsible for leading human genomics. And so my role is to lead all aspects of what the institute does, both with respect to our extramural program, which is our grant-giving program that helps fund research in human genomics in the US and even abroad. But also we have an on-campus, what we call an intramural research program that also pursues human genomics, everything from very basic genomics to genomic medicine.
Lovely. And Dr. Barwell, would you like to introduce yourself? You're coming from the UK.
That's right. Thank you so much for the invitation. It's great to see you again. So I'm a clinical geneticist, and my main area is familial cancer. So we see a lot of people who are either affected with cancer or at risk. But we've also been moving more into personalized medicine. So we're thinking about how we can best treat people mainly for cancer, but other conditions as well. And I've got an honorary professorship in genomic medicine here in Leicester, which is in the middle of the country. And what we're trying to do, apart from teach people the difference between genetics, personalized medicine, and genomics, is create that vision whereby how do we inspire the next generation to think about screening not purely based on age, think beyond 500 years of the microscope, and actually give people the right drug first time at the right dose. And those are the sort of the three pillars that we're trying to inspire and enable and empower the next generation to take on board and to bring together different data sets to make better risk predictions. So thank you very much.
Yeah, that is amazing. And Dr. Brinkman, you're coming from Canada.
Yes, I am. Thank you. It's a pleasure to be here. I've really had a long time interest in sort of bringing data together, and got interested as first a microbiologist, and then increasingly in genomics, in data integration, and started to build the child cohort study database here in Canada. This database is got about 57 million data points of incredibly diverse data on these children, including genomics data. Excuse me. And what it's allowing us to do is really identify these through the child and the parent trios. We're really able to identify the impact of genetics and environmental exposures on health. And the goal is ultimately to be able to better understand these trajectories children have. They may start with a bad deal in life, or they may have a better deal in life. But what we want to do is really improve those trajectories so they're all ending up getting the best start they can.
Yeah, amazing. Well, I think that gives us a really great baseline to go off of to start our conversation of just where all of your expertise lies, and kind of a teaser of the topics that we're going to dive into. As we are starting this conversation, I know it's kind of a silly question, but for those that are maybe newer to this concept, I think we should define what population genomics is and how we approach that study, how it's currently being applied in the clinical practice as well. And if you have thoughts on how it's changed, in recent years. I don't know if anyone has thoughts that want to get us started.
Kira, maybe we could also make sure everybody understands, or we have a firm grasp on what the difference is between population genomics and maybe genomic medicine.
Yes, I think that is beautiful. Dr. Green, do you want to answer that for us?
Well, I will happily answer it, but I will readily admit I'll give you my answer. And I actually
yes.
think, we have some experts who think, all three of us, I think you're going to look at this all in a slightly different way. Genomic medicine, let me start with. And genomic medicine to me is using genomic information about individual patients to manage their medical care. So I think probably we will mostly agree with some wording like that. Population genomics, which actually is just I think a reinvented term from population genetics, which even the word genetics is a much older word than genomics. I think historically has had significant evolutionary and connotations. But I think where our discussion is going to go, for where I think you want to have us discuss, is some of the diagnostic implications of population genomics. So to me, there is a Venn diagram, but I think there's a lot of aspects of population genomics that sit out, that are more about evolutionary biology and human migrations and lots of things that have incredible, interesting topics. I suspect this conversation, you're going to want to focus us more on what are the implications of population genomics when it comes to diagnostics in the context of genomic medicine. That's
exactly
my take. But I'd
yes.
be curious if my fellow panelists agree with what I just said, and I'm sure they could add to it.
Yes, and feel free to disagree with each other. It always makes for an interesting episode here. But Dr. Barwell, Dr. Brinkman, any thoughts on what Dr. Green was sharing?
So I'm always struck as we move into September and I start to dust off the Christmas Carol, a great British story from Charles Dickens, and we think about the past, the present, and the future. And for me, genetics is a study of our biological clock. And people ask how old I am. I say genetically, I'm 2 billion years old. That's how old and sometimes I feel it, I really do. And then when I think about personalized medicine and we're trying to get away from thinking about the reason for a particular medical issue, we're trying to understand the response. So that's where microbiology and tumor genetics and pharmacogenetics fits in. So that's about the present. What are we going to do now for this particular patient? And for me, genomics is how we start to predict the future. Rather than stargazing into the past and time traveling, we're crystal ball gazing. And that involves bringing together your DNA code, but also the other aspects which are just as powerful, such as your postcode. And how do we integrate? So linking into the sort of things that Fiona has been talking about, how can we make better predictions based on the different data sets? And the population aspect of this, for me, is what do we actually know about data outcomes already? And I think the area we sometimes forget about is that linking not just of the data sets that we have, but tracking them over time, because that provides the opportunities to actually find those key patterns, not just in evolution and migration, but actually start to make better healthcare and well-being predictions as well. And that opens up all sorts of ethical issues about disparities and also about how we use data and store it.
Yeah, I think that's a great point. And Dr. Brinkman kind of started us on that point during her introduction of talking about just data sets and how important it is to have a diverse data set. Because if you don't have a diverse data set, we're not going to be able to use that data to then properly have that in the system to then pull from when we're trying to diagnose for certain genetic disorders or screening and all that. Dr. Brinkman, I would love to hear your thoughts on those topics.
Yeah, thank you for this. This is something near and dear to my heart because we've been analyzing this in Canada, trying to get cohorts and for analyses that reflect our population. And it's challenging. It's really challenging to get at this
access into research projects through, for instance, the Clinical Research Network, which is through something called the National Institute of Health Research. Our links with organizations such as Public Health England, Genomics England, provides great strengths and opportunities and integrated databasing. The challenge is not in the strategy or the standardization. The challenge is in the capacity and delivery, because the number of tests that are being available now, and not just in terms of thinking about classical genetics, but thinking about personalized medicine and moving into genomics, means that there are huge information governance issues and workforce management issues, because to create those scientists that understand the data and can help curate it and to help us interpret it, it takes years. And I'll stop with just one thought, which is, I heard someone say that 1 in 3 jobs for our children at school don't yet exist. So we're trying to teach and train our young people for things we don't even know about, that haven't even been invented. And we're moving into the world of AI and data integration and new sciences. But the reality is, you've got to predict and take a punt as to where the opportunities are going to be in the future. And training and attitudes towards change are going to be really important. So workforce planning, I think, probably is a #1 challenge for us here in the UK.
Yeah, that makes sense. I imagine that the 3 of you have experienced that over your careers, of what you started learning in undergrad, grad schools, and your PhD programs and everything is very different than what you're doing today. You guys have really experienced.
The word genomics did not exist before 1987.
You're so right, Dr. Green. It was genetics, but
it was genetics, which was coined in about 1903. And genomics did not exist in the medical or scientific literature until 1987. I'm not going to guess the ages of my fellow panelists, but I will be very bold on saying I graduated from medical school and graduate school. I had my MD, PhD given to me in 1987, which means never once during my formal education, grade school, middle school, high school, college, MD, PhD, never once did I hear the word genomics.
Wow.
And yet, so this, I think what Julian said is spot on. And I'm not even sure the phrase data science existed more than about maybe 10 years ago. And now there's not enough data scientists in the world. So I completely agree that the workforce of the future are going to be experts in areas that haven't even been named yet, just like all of us, or at least I have become an expert in a field I never heard the word once in my formal education.
Yeah, no, that is a very astute point between the 2 of you, that it's just things change very fast in genetics now, genomics, with all that. And Dr. Brinkman, I kind of wanted to pull you in terms of the Canadian perspective, because from the little I know of, being in grad school with Canadians, that's kind of how I've learned the most of it. It's similar to the UK in terms of being under one umbrella, that the government really has this healthcare system. But can you either debunk what I just said or kind of explain that further?
I actually can debunk it a little bit. So first, I'll just mention, just to step back, that I absolutely agree with both of you about the sort of
like the workforce issue,
development. But I will disagree in the sense that I think what's key is to develop good critical thinkers, and then they can be agile and handle. You guys have done very well in your careers because you probably do have very good critical thinking and social skills that were necessary to basically pivot to whatever comes up. And I think that's what's going to be very important in our school curriculum. I would really love to see more emphasis on that. And the fact that, yes, you do not, what students are learning right now, they are going to be doing something different in the future, and that's okay. They can pivot if they've got the right tools and skills. So, but I also just want to say that for Canada, it's actually a federated healthcare system. Our system is much more complicated than we're very jealous of the US and the UK because you have some really nice infrastructure, some nice setups that basically we do not have in Canada. We depend on the UK and the US for many aspects of our genomics, from databases to several analyses. And the federated healthcare system, which means each province and territory is their own healthcare system, can be a challenge. However, it's also a real plus, and that is that what we can develop in terms of our federated healthcare system, in terms of data sharing, in terms of genomics and coordinating that, that can be a model of what can be done on the international stage. And so we have initiatives like the GA4GH standardization, where Canada has played one of the lead roles in that, in part because we're just so attuned to this issue of the importance of data standards, data harmonization, and getting organized when it comes to a federated system. So, hopefully we can really make some inroads in that front.
Yeah, yeah, definitely.
I just point out that everybody's always jealous of the other healthcare system. I mean, because
it is true.
Fiona, think that the U.S. healthcare system is something to be proud of. I'm not proud of it. I mean, I'm proud of our advances. I'm proud of our capabilities as a comprehensive country country view. We have a lot of problems. We're a terribly broken system at many levels. There's another dimension, Kara, that I think we want to make sure to emphasize, that I think each of our countries, just like every other part of the world, also faces significant differences with respect to the diversity of their population. And that brings in, which relates to this discussion about population genomics. And certainly the US is this remarkable melting pot. And I'm not claiming we have the biggest challenges associated with diversity, but I think our challenges are certainly different than what they face in the UK or up in Canada. And again, it's not better, not worse, not harder, not easier. It's just different. And so I do think that some of the things we deal with here, both for healthcare and for cohort-based studies that feed into population genomics topics, it's just a different circumstance. And so that is also another variable that each of us deal with, both on the research side and on the clinical side.
Yeah, no, it definitely is. I think all of them have their perks and their consequences, but grass is always greener on the other side, right? And when I'm talking to my Canadian friends, it's like, oh, well, they get this covered and this covered, but the wait list might be really long compared to, if I'm seeking healthcare or something on different aspects. So there's always something that's better somewhere else. But I think you guys all highlight really interesting points and just those strengths and weaknesses of the different healthcare systems and what we can learn from each other. I think it's interesting when I've seen, one of the areas of like genomics that I've been following is a lot of these baby seek projects, where in the US, in certain states, we've been doing, like, I think it's baby bear in California, baby deer in Michigan. So there's these different projects, but that it's, and Dr. Green, correct me, because obviously you know this stuff better than I do. But it's interesting to see these studies coming up, but then comparing it to, like, in the UK, you guys have had the 100,000 Genomes Project, and that's been going much longer from my understanding. And that with the kind of healthcare system you have, it's easier to be organizing all of that data, because in the US, from my perspective, we're kind of fragmented because we don't have one centralized database. So I imagine just when it comes to data sharing, it's so much easier in the UK and to some extent Canada compared to the US. Well, I don't know. Am I getting this right? What do you guys think about that?
I mean, my job, I'm constantly herding cats. I mean, that's what we have to do. It's like a bunch of different cats, all going different, and getting the data to be shared and having any sort, there are things we can do when we fund grants to sort of help to coordinate it. But I live in jealousy of what's going on in the UK, because somebody high up makes a command decision, and because of the way their healthcare
filters
system,
down,
boom, there they have it, and they've got a lot of zeros after their number when they got to 100K. And the fact is, we are eager to see their results, and we're going to take notes when they report them, because we're going to learn a lot. I think
exactly
has really greatly made them the leader in many of these things, and I think it's terrific, and I applaud them.
Yeah, and I think that's why it's so important that in this age of, especially, I'm sure we're going to talk more about AI as well, that we're just able to share so much. I think much more than decades ago when it was much harder to be working internationally with people, just with the technology that we have today. To dive in a little bit deeper into more details and examples, would love to hear about how you each envision genomic data being effectively integrated into the healthcare systems to then benefit patients. Because at the end of the day, the reason we're doing all of this is to benefit people, right? We're trying to make healthcare better and have better outcomes for people, whether that be preventative medicine or personalized medicine, or, a mix of all of these different types. But maybe just thinking about an example of a patient walks in, how were they affected by the behind the scenes work that it took to get there? Anyone have thoughts? I don't want to pick on someone right away.
Yeah, so for me, the main win in medicine will be pharmacogenetics. That's the low-hanging fruit. How can you get somebody in accident and emergency on a Friday night, or your general practice on a Tuesday morning? How can we get to a point where they're able to get that right drug at the right dose and reduce harm and potentially improve compliance? And there's a logistics around getting that test. There's also a training issue about, because none of the doctors or pharmacists would have been taught about how this is going to be implemented into clinical practice. And then the third thing is patient-based solutions. So how can we get to a point where you can QR code your prescription and get that red, amber, green, simple signals? So I think that's the first point we will start. Rare diseases, there's several thousand. And so although we could pick off some of the more common ones, or the ones where there's multi-system aspect to it, and of course you look at familial breast cancer with computer modeling, and PhenoTips obviously can help with that. By bringing in different data sets such as alcohol, body mass index, etc., as we link across to CAN risk, for instance. That's going to take to the end of the century before they're absolutely driving it. But I would argue that in the 20s and the 30s, we've got to start giving them the tools as well. And that, because it's the patient who needs to own this really in time.
Yeah, I think that's a good point. And that we've seen pharmacogenomics and we've been using it for years, but I feel like we've been dabbling and it's not becoming a standard in terms of in healthcare practice of, all right, let's check this. We're going to give you're coming, as you kind of said, to emergency care, like an ER, an ED, and you need pain medication immediately, right? Because something catastrophic has happened. Being able to have that data on file of what are with your genetics, is it better to give you drug A or B? And then what dose does it make sense to give you? And that's information that immediately could be provided in affecting the patient. Obviously, way other areas, right? Because pharmacy is just in every area of medicine. But I think that's something, because we've already started using that, especially I've seen that used in the psychiatric area of what med is maybe best to start you on for depression, anxiety, different mental health challenges there. But I see this as expanding very, very quickly there. Dr. Green, Dr. Brinkman, other kind of thoughts?
I like that optimism that Julian expressed. I especially around pharmacogenomics. I used to lead with pharmacogenomics when I would sort of rattle off my list. And I will tell you, at least here in the U.S., there's been a little bit of pushback about maybe that optimism isn't being matched with reality. I'm hopeful that, and maybe some of the things that need to happen that Julian mentioned are exactly what's holding us back. But I've been disappointed with the uptake, and I share some of the barriers I agree with that I've heard, and I'm hoping we get there eventually. But I in my more recent conversations, I tend to list it about third or fourth on my list because the uptake hasn't been as aggressive as I had been hopeful. By the way, I will point out one thing we didn't mention. So we touched on rare diseases. I think we touched a little on cancer, which will probably be a big one. We certainly, I certainly agree. Pharmacogenomics, the potential is tremendous. But let's not forget non-invasive prenatal testing. They always lead with cancer because I think in the long
able to collect and organize all of this information. So, I mean, Dr. Green, as the director of the I have to get the letters right, NHGRI, how is your organization working to ensure that we do have optimal utilization and collection of the genomic data so that we can effectively use that in clinical applications? And then Dr. Brinkman, I'm going to want to hear from you after about just like sorting through all of that kind of data, because you have like a specialty in that. But yeah, I mean, for being in the US, especially with all of our fragmentation, how do we even approach that? Like, I'm even thinking about, you know, the All of Us research program is massive. Like, how do you even approach this to make sure that we're doing this in the best way we can?
Yeah, no, it's a great question. There's a lot of things packaged into that question. So the first thing I want to distinguish is reference genome data. And I want to put in a request now that we come back to the pangenome, because I think there's a really important point about the pangenome that relates to this conversation around population genomics and diagnostics. And I want to make that point. So one thing we do is we are funding the creation of a lot of genome sequences from diverse populations and packaging them within this pangenome program. And I want to come back and talk about it. Those are reference genomes. But I think your question was probably more along the lines of how are we collecting all the genomic data that's out there, both in terms of genomic data from whole genome sequencing efforts as part of cohort studies or disease-specific studies. A cohort study is being signed like All of Us, but there's a lot of other data that's being collected associated with lots of other studies. And then, of course, there's also the data that's being produced by diagnostic companies about genomic variants and their relevance and linkages to specific phenotypic information. So those are, you know, there's a lot of different answers for all these things. What I would say is the NIH, starting with our institute and having a very strong data sharing policy that if we give you money and you generate genomic data, you must share it. It has to be properly consented. We have to put it under controlled access because it's human data, but you must do that. That then got generalized to an NIH-wide policy, such that now that is sort of the expectation, with some minor exceptions, is that you must share your data. And then if you look at the All of Us program, the All of Us research program, and 1 million individuals, you know, that's actually, you know, that's absolutely a term of enrollment. And, you know, the vast majority of individuals are certainly allowing their data to be shared, and that will eventually yield over 1 million genome sequences or more from people in the US. When it comes to diagnostic companies, it's up to them. We didn't pay for it. Government didn't pay for it. Healthcare paid for it. And some are being altruistic and are sharing and putting it in data resources, and others are not. And there's all we could do is arm twist, but we can't control. But I would say there's significant commitment at NIH to get this data shared, develop the infrastructure for housing that data, having a lot of software and computational biologists, data scientists analyzing that data, developing resources of these genomic variants, and eventually through efforts like our ClinGen program, Clinical Genome Research, Clinical Genomics Resource, or ClinGen, is this idea of getting people to better curate and understand what the role of these genomic variants are with respect to gene function and genetic diseases.
It is really hard when we see that so many, like when tests are ordered, like let's say I have a patient sitting in my office, right? And we're ordering testing that is being sent to a lab that is a commercial lab. So they are keeping that information. I'm not sending this to the NIH. You guys don't get that information. So it is hard because you've got so many labs that have their own databases. And that's why, you know, as a clinician, as a genetic counselor, we have this tough thing where we're like, well, one lab calls it a pathogenic variant. One lab calls it a variant of unknown significance or uncertain significance. And we're like, well, which one do we go with? We don't have this centralized database to say, oh, well, this is what us as a genetics community has said. This is what this variant means. So I think that that's what's tough, but I'm starting to see changes of some major labs that have kind of been under fire in the past for not data sharing, making big efforts to data share. And I don't want to name names, but you know, I think we're all kind of thinking of the same ones, but it's encouraging to see that data sharing is happening, but And it's hard. You can't just data share all at once because these are huge, huge data files. You can't just be like, oh yeah, here's 1 million genomes. Here you go. Let me just airdrop that to you. So I think there's also just that administrative side that's really tough. But Dr. Brinkman, when it does come to, analyzing these variants and all of these big data sets, I mean, how do we even start to do this? Like, I remember you mentioning at the very top when you were introducing yourself, something like, was it 57,000 million like data points? Like, I mean, how do we even start processing this? Are you starting to use AI within your teams to do this? Like, to me, as someone that's not a biostatistician, I'm like completely overwhelmed with that number. I don't even know where you would start.
Yeah. So, yeah, I think there's a couple of things here. First is that it's, when it comes to the sort of quote contextual data that, is a lot of those 57 million data points is, that you often have to start with coming up with what we call a minimal metadata model, like this minimal information that you think people should share, like they should all be sharing sex and describe. It's amazing how many different ways somebody can write down male and female and other, it's just amazing. But basically, if we can just start to standardize some of the basics, and then you build on that with the Seneca project, which is a project that was doing cohort data sharing between North America, Europe, and Africa. We found that another thing that was really helpful was actually synthetic data. Is if you want to build these resources to help with developing ways to be interoperable and share data, just coming up with some synthetic data sets so that the developers of these infrastructure can sort of see what examples of what's being done without actually using real data, which might be subject to difficulties in retrieving it from certain countries. So we found that was really, really helpful. But so in short, I think there's a bunch of things that can be done. But we still certainly have a long way to go with regard to getting really organized and getting to that point with, just standardizing approaches and methods. And I think one challenge is going to be that this is a moving target. The technology is going to gradually keep improving, and what we collect is going to keep changing. And so it would just be nice to make sure that globally we're setting some resources and infrastructure to sort of get organized, because it's like garbage collection. Many companies have learned you sort at the source. You sort at the source, you get organized at the beginning, and then it's much easier. If you try to sort of sort or do things after the fact, after you've collected massive amounts of garbage and then sort it, that's much harder. So yeah, there's a lot we can do, and not to imply genomes are garbage. I think it just, but there
might be parts that are less important though.
Yeah,
yes. Yeah. Trying to streamline and say, where do we need to focus maybe for this particular study versus another one? And yeah, just having all of that data kind of in the same format, like you said at the beginning, like people might answer a question very differently, and you have to, make sure that that's a binary, and there are only maybe 2 answers or a set number of answers that could be said. But I wanted,
if I could I will add that regarding the AI point, I think there's a lot that can be done in this area, but we do have to be very careful. We do know that some of the AI, like the large language models, they do hallucinate. So I do think it's important that we use that. For example, I found it most useful for creativity and coming up with ideas for things. But when you're wanting to really generate hard data, you can use something like that too. Like people are using it to help with coding some things, but you really do have to be very careful still with this. It's still in its infancy. But I think there's a lot of potential there, and certainly been very encouraged by some of the AI developments in some areas with regard to medicine. It's been really impressive what's happening.
Yeah, it's Fiona, been exciting.
Has hallucinate become a technical word in data science?
I saw you laugh at that, Dr. Green.
I've never heard that said that way. I like it.
Sorry, which? The hallucinate?
Okay.
Yeah.
Yeah.
No, it's an official term. It's an official term.
Is it? Wow.
Yeah, like ChatGPT hallucinates, and that's and I've seen that happen. I mean, it's pretty funny when it comes up with something completely.
It kind of it tries to fill in the gaps. And sometimes the way it does that is you're like, I don't know about this, but I agree with you. Even in my own life, I think it's been really great for idea generation and like writing of like a draft. But yeah, I think it's also important for people to, they're thinking about, oh, how can I utilize AI in my own workflow of stuff? Make sure you're not putting any HIPAA information into that, because if you're not using your own AI software with your company or government, then I don't know, it's kind of a scary area right now. And I wanted to mention that we do have some questions coming in. Would love to see more questions. I have plenty of questions, but I want to make sure we answer all of yours in the audience. But before I get to those, Dr. Barwell, I did kind of want to ask you, is someone that kind of, works with patients and everything. You mentioned at the beginning that it's going to be important with all of this information and really being able to utilize like population genomics in clinical practice, literally the name of this installment here. It's going to be important for patients to be taking the lead on that, and for people to say, okay, I'm doing this education for myself and I'm teaching myself what I need to ask, and really being their own advocate with it. But that means they're going to have to learn a lot of this. And, all of you have had really successful careers in genetics, and obviously you've had a lot of training in this. But for other people that are not in genetics, not in healthcare, how can we really be educating people and engaging them in all of this genomic information and having them understand the implications for healthcare? Because I think this is something that, science communication is always an area that we need to focus on, but genetics is getting, and genomics, I should say, is getting more and more complicated. So I think it's harder to teach, if you're watching a news show or you're on NPR and you've only got 6 minutes. What are your thoughts on just how we can best inform the public?
Yeah, so I'd split it into two. So when I started in clinical genetics, you had to be able to say, well, that patient, I remember I read a journal about this, which was published in 1978, and we need to go for this gene, and it's going to be this variant, because testing was expensive and it took years. So now, rather than what gene do you think this is, the question is, we've got all this data, what does it mean? So it's therefore really important we get very good, not only genotype databases, but also make sure that the clinicians are putting in really nice phenotypic data, and that we get the data sharing that we talk about. So when it comes to individual variants, it's going to be linking those data sets around the world. And I'm really struck that if you've got somebody from Africa who's managed to get into one of our clinics versus somebody that is native to Leicester, for instance, even when you've got them and they're putting out their arm, the chance of making a diagnosis is so much lower because of those disparities of the controlled databasing that we've talked about. So disparities are multi-layered, culturally, language, but also at a genomic level. The second issue is really about whether we move into a world where we're almost going to have a digital twin that we can use for modeling if we get cancer, digital twin, decide on what drug we might have, but also to think about our cardiovascular risk. If I have these 6 donuts and don't go to the gym and don't buy that dog, and my blood pressure is this, what is it likely to do to my quality of life and my length of life? And of course, the second point, if you're happy to generate that digital twin, is, are we going to be comfortable with the decisions that that computer model comes up with? Is it a case of computer on, brain off? And are we going to be comfortable to link our results to our DNA code and our postcode? I mean, in 2020 in the UK, we had A-level results, which are the exams that I guess similar to the end of high school at the age of 18. And they were linked to previous attainment, but also the school's attainment for the last 5 years, effectively linked to postcode, where you lived, your zip code. And a lot of people were very unhappy with that because it was outside their sense of social justice and control. So digital twinning and better predictions is one thing, But we are going to need to think very carefully about, do we feel that it hits into our sense of social justice when it comes to delivery? There's an issue with regards to the collation and the accuracy, but a whole nother ball game when we think about how you might use that data. Once you're linking in your food choices, your Fitbit, your sleep app, if it tells you your risk of Alzheimer's is 44% higher, what are you going to do with that? What happens if your insurance company gets access to that, or your health insurance? So I think there's a lot of issues about how we integrate that data and then how we use it. And I think we need to be almost thinking that the technology is not going to hold us back here. It's going to be how we respond to that as well. And I think we need to urgently have that debate as well.
Yeah, I think that's a very good point. Dr. Green, add on to it.
Yeah, well, what I want to do is I want to pick up on the first point Julian just made related to disparities related to the genomic data, because I think it'll tie back into pangenome.
I was going to say we had to hit that.
But ties into population genomics. And I really want to make a very important point that I found was sort of an inference that some people struggled with. And I mean, some people meaning the general public, or maybe even some of my colleagues. This relates to earlier. So keep in mind, this is what gets to be very important, because if a little bit of sequence from a patient's genome does not match anywhere in any of your available references, it gets tossed out. Most of the time they don't know what to do with it. It sort of like falls on the floor because it doesn't fit in anywhere. And so this is why we need to have a set of reference genomes. We need a pangenome, so that every person's genome who we sequence as a patient as part of genomic medicine, we will make sure to capture all the sequence data being generated and know where to put it in the genome and know where to contextualize it for diagnostic purposes. And so this is why this idea of a mismatch between the references that are available and the diversity of patients that walk into clinics becomes a disparities issue, which is one of the things we hope to solve by creating a human pangenome reference. Now, that sounds easy, and there's a million questions you could ask about how exactly is that going to work, and how do you match them? And all of us, many people are blended, and so we all have two genomes in us. All great questions, which is why at least we and others around the world are funding researchers to figure all this out. But I think it's an important thing to understand is that without adequate diversity among our references, some people's genome segments may never be analyzed appropriately. And if they contain medically relevant information, that information will be lost.
Yeah, that's what I was going to say of like, let's say you have a kid with an undiagnosed, what we think is a rare disease that has a genetic cause to it. And it is a single gene or a single nucleotide change where you have an A changes to a T, something like that. But it's in that section of the genome that is on the cutting room floor. You're going to miss that diagnosis.
Exactly.
So yeah, so that's really concerning. I didn't know that that's how that worked until this moment. So I'm sure other people are having that same aha moment I'm having of like, oh God, that's how it works.
And remember,
So,
and it's a really important point, a diagnostic genome sequence is, reasonably inexpensive. It depends which currency you're using, but, less than $1,000 US. A reference sequence, high quality, where we get every last nucleotide, is tens of thousands of dollars. And eventually we hope will be cheaper. But again, what patients get for genomic medicine is a quick and dirty, cheap genome sequence. It totally relies on a properly matched reference to make sure things don't get missed. That's the
wow,
reason why we need a pan-genome.
that is so important.
Among other reasons.
Right. But that being a major reason when it comes to diagnostics, especially for people that are coming from different areas of the world. Yeah, that is so important. Wow. I hope if people are tuning out, rewind the past 5 minutes and listen to that again. But we're going to get to some audience questions now. So our first question is, how does the panel imagine first contact general practitioners who typically are less trained in genomic medicine? How can they effectively participate? And how that's going to work? If I'm going to the doctor and I'm seeing my GP for something, how are they going to take advantage of genomic medicine just being a general practitioner? Because a lot of what we've been talking about is in different specialties. We've talked about pregnancy. We've talked about cancer. We've hit on rare diseases, cardio a little bit. So what about just general practitioners that, as we've mentioned, especially if they're of a certain age, they're not going to understand genomics. And even if they're younger or they recently came out of med school, they only learn so much genetics. We have medical students that rotate through our office, and I love asking them. I'm like, so how much genetics have you learned so far in their third years? And they're like, not much, which I hate hearing. But what are your guys' thoughts on just how general practitioners can take advantage and start using this in their own practice, or what they should be aware of?
I will just add that I think one thing we have to be very sensitive to is that the general practitioners really need interpretation and tools, because they're really expected to know a lot. And we should be, I think we have a responsibility in these different areas of genomics and different applications of genomics to really provide that information in a good, robust, digestible form. So I would really like to see more of that. So I think it's a little bit more, the onus is on us to really fix that up.
Yeah, just more resources.
The professional societies are going to play a very important role here, especially among the specialties. We help support and stood up an organization called the International Society for Practitioner Education, and I'm getting the letters wrong. The ISCC is the coordinating center, and then it's Practitioner Education and Genomics. It's basically an umbrella group that it helps pull together different specialty professional societies to help develop the proper educational materials, working with us, working with others, to stand up the right kind of information they need to be able to implement genomics in their clinical practice, recognizing there's many, many nooks and crannies of the medical ecosystem.
Yeah, definitely.
So for me, there's about 4 different main presentations. So you've got the patient that comes in with a large report, which has got 35 different genomic variants, hemochromatosis, alpha-1 antitrypsin, BRCA1, and they're unsure which one's significant because it's intelligible to the patient. And that's a real heart sink, I think, for the GP. And I think when you've got something of that complexity, it's almost certainly going to have to go to clinical genetics. The second one is something like a SNP analysis factoring in cardiovascular risk or breast cancer risk, a computer model for a particular condition. The third one is they come up and they say, oh, my daughter's just got this gene report, which says she's got this particular condition. And then the classic one, which is the patient who's got vague abdominal pain and they're feeling a little bit tired. And the critical thing is to ask, well, do you have a family history? and you find out that it's likely to be Lynch syndrome because of the bowel and gynecological, and you've missed an opportunity, if you didn't ask that, to pick up the right-sided bowel tumor or the endometrial cancer in someone premenopausal. And so, first of all, most GPs will learn through those 4 types of case presentations.
Yeah, yeah, definitely. And I want to punt this next question to Dr. Brinkman. With the volume of data presently available, what is most needed to practically leverage the data towards improving healthcare outcomes for patients? I know we've talked a bit about this, but anything else to add just when it comes to actually utilizing the data?
Yeah, so I think it's, again, a bit about getting organized. At least that's certainly one thing I see. I think also this issue of diversity is still really important. Even people who, we really need to properly address the pharmacogenomics challenges through this idea of having diversity, so that we can really make sure this is going to be effective for everybody, not just effective for 80%. So certainly I would say, it might seem counterintuitive, but just in general, like, everybody will benefit from more diversity to make sure that we deal with the fact that essentially nobody's sort of common. I always love that comment about rare diseases. I mean, they're so common. There's so many that they're rare diseases are really not that rare. And so, and likewise for all the different kinds of idiosyncrasies your particular genome might have. I think we really need to make sure that we are capturing that diversity. And but really it does come down to getting organized. I would also like to maybe add the sort of social sciences side of this. We tend to focus on the science and the technology, and we come up with this great idea for a test, but then we've got to make sure that test and the way it's being implemented really works with society and how people are going to interpret it, how people are going to feel when they get that result. We see examples of people getting a genetic test where it's really beneficial that they learn about something and can get a treatment. You get people who get a genetic test and they learn they do not have a mutation that's found in their family, and they have a bit of survivor's guilt that they don't have this thing that other people in their family have. So I do think we do want to be really careful, and getting back to that idea of community, that there's sort of protecting individuals and what they their rights, but let's not forget about that some of these impacts can be at the sort of community level, or involving certain populations of people that basically this a particular issue is affecting.
Yeah, I think that's very well said, and I think we have time for one more question here. And Dr. Barwell, you'll probably be the best person to answer this. Recently, the British Society for Genetic Medicine published a guidance for clinical practice about the retention of family records in genetics. How do you see current clinical data retention policies impacting the way in which genomic medicine and population genomics is delivered in practice?
So it is changing to a degree. Genetic counseling was very much based on the Huntington's model. Why would you want to know? And we must be protective of patients and their mental health and family dynamics and who has access to this. As we're moving to a more personalized medicine and more mainstreaming, where these tests are carried out more broadly in healthcare settings, we need more people to be able to access that result. But there are major implications, because obviously you need families sometimes to come together to make a diagnosis, and yet everybody has a right fundamentally to their own confidentiality. So the middle way is that pedigrees will have to sit behind a firewall, particularly if they've got names. But the actual genetic result of the patient will be placed into a more generic model of electronic healthcare record. And that's something that we're starting to see more of in the UK, particularly for personalized medicine. I go further and would say, well, actually, we don't have the health record. The patient has the health record and releases it to me if they look at me in the eye and think that I might be able to be trusted. And so I would change it even further. But I think we're moving in the right direction. Obviously, it's always good to be cautious and have debates about these things, because, as you say in families, once you've said something, let the genie out of the bottle, you can't bring it back. So we have a national genetic ethics group which discusses these issues, called Genethics. And I think that's a really great resource. And it's genetic counselors who are very much leading the way here.
Yeah, definitely. I think that's a really good point. And just, I like your idea of patients being able to release their own data to specific healthcare providers. I think that would be a great thing to be able to accomplish. I think
see it be relevant to others.
Yeah, I think that's very well said. And to close us out, because we've come a long way in the last 20 years. And this is the anniversary, because it was 2003. So we are in 2023. So now it's been 20 years since that first draft. And we've made so many improvements since then, even the telomere to telomere, final project that was, I want to say, maybe April of 2022, something like that. So it's just remarkable how far we've come. And I just have to thank you so much, Dr. Green, Dr. Brinkman, Dr. Barwell. I mean, this has been such a fascinating conversation, and I've thoroughly enjoyed it, and I've learned so much. I'm gonna go out and say it's one of my favorite installments of the PhenoTips Speaker Series. So I think this has just been such an awesome conversation. I had high expectations. You guys exceeded them. So I just really appreciate hearing all of your unique perspectives, especially coming from 3 different countries. I think it's just remarkable that we're able to get together and have these conversations and see what's different, what we can pull from each country's healthcare system, and the type of research that we can do with either the advantages of that, the disadvantages with limitations that we have in certain countries, but really just coming together so that we can continuously, start using population genetics in clinical practice. So thank you so much. Everybody, as we close out, you'll see a feedback link in your browser when this webinar ends. And we're also going to email it with you, to you in the next day or so. So please take a minute to fill it out. If you want to hear more topics like this of population genomics, ways that we're using genomics in clinical practice, what other topics you want to hear about, we would love to be able to plan those installments in the near future. You can also sign up for alerts on our upcoming sessions so that you can participate in them live. We also, if you do miss them, we do have it available on our website, phenotips.com. If you hover over the resources tab, Speaker Series is going to pop up on the drop-down menu. All the installments are on there. You can also check it out on your podcast app, Spotify, Apple, Google Podcasts, which is now becoming YouTube Music. So just search PhenoTips Speaker Series, will pop right up. So yeah, stay tuned for our next webinar. Thank you so much for everybody for tuning in and everyone's attention, and for the 3 of you for just being an awesome, awesome panel. Thank you so much. Have a great day, everybody. Bye.